Okay, welcome to new session. Next speaker is I would like to introduce Nicolas Clercin from Paris, France. He's very interested in algal ecology, test and other problems. Now he's going to talk about behavior of the test and other compounds from the outbreak of the breakthrough. Nico, the floor is yours. I think enough for you introduction.
All right, thank you for the gentle introduction. All right, so can you hear me well and see me? Because I don't see myself, I see Marriage instead. I assume so. I'm currently a researcher at École des Ponts in Eastern Paris University. I study basically water quality in urban lakes, and I'm really interested in algae, phytoplankton populations, dynamics, and organic matter production associated with algal growth in urban systems. So today I'm going to talk a bit about the behavior of taste and odor compounds we can see in some surface water, drinking water supply reservoirs, and the title today will be Behavior of Taste and Odor Compounds from Outbreak in Real Systems to Breakthrough. I cannot switch the slide for some reason. It's not working. Rita? The slide is frozen.
Yes, yeah.
It's not showing up. No, we see Marriage. Marriage, maybe if you turn your camera off. Active.
Okay.
Okay, I have to switch from these arrows. Okay. Okay, can you see the next slide? Yeah, okay. Okay, good. So taste and odor are a real issue, so well-known issues worldwide for water company, for water producers or water managers, because taste and odor outbreaks, they taint the source waters and they generate foul smells and sometimes very difficult to remove with the traditional water treatment processes. And of course, because the finished water at the treatment plants, if it smells or tastes is funky, can be a source of complaints from consumers. Because aesthetics of the water are the primary criteria that determine the confidence of in a water supply. What to say? The water has to taste well. Depending on the perception of individuals, we have our personal perception of what good water tastes like. So if it should be have no flavor, but it does have some flavor, but we are less or more sensitive to those flavors, and especially the color of the water is probably the most important criterion determining a good water quality. If the water is slightly tainted brown, you may not drink it because there are some suspended particles in it. And also, so yeah, those taste and odor compounds are paying for water producers and also in fish farms because it costs a lot of money to remove those compounds from the water. And in fish farm industries, they bioaccumulate in the fish flesh. Sorry for the slide. The next slide, maybe. No, it's not coming. Oh yeah, yes. So this is a 10-year time series. I said non-fictive reservoir because for data disclosure, I'm not supposed to mention the study site. And but this is very interesting because on the top panel, you can see the dynamics of one of the compounds, MIB, 2-methyl isoborneol. And the bottom chart is geosmin in red. And as you can see here, the dynamics of either compounds is highly variable between years. So some years we have bad taste and odor issues, and some other years we don't have any problems, or very, very low detections of these compounds. And so I like this chart because it really shows us that the duration of a taste and odor event can be variable, and usually MIB in my study site, MIB occurrences are shorter than geosmin occurrences in the raw water. And of course, we have the feedback sound. Of course, we also have variability in the intensity of the taste and odor concentrations. It can go from beyond the other threshold concentrations to several hundreds, and sometimes in bad cases, we have can reach a few thousand nanogram per liter of like it's the case in 2014 in geosmin. Like if you see my cursor, I don't know, this spike was switched. This spike was several thousand. So, well, basically, taste and odor analysis in the current study was analyzed using GCMS, HSSPME GCMS method, and what we did for some years, we duplicate the samples to know the fractionation of particle bound or dissolved compounds, or either dissolved MIB or geosmin, and but the filtrate were analyzed the same way that the raw water samples. So we were able to determine the particle bound. It's just a simple relationship to the total concentration minus the dissolved fraction analyzed with the GCMS. And if I can switch, sorry. And this is what we got. So these two charts represent in light color is the dissolved fraction, in the dark areas represent the particulate or intracellular concentration of the compounds. And as you can see, it's variable. Geosmin, for example, is most of the time, we'll say, it's more likely intracellular, whereas MIB is most of the time dissolved fraction. So this is it's resumed here in this small table. So we analyzed more than 1,300 samples. So 96% of these samples were positive for geosmin, and 79% were positive for MIB. Out of these samples, 76% were exceeded the other threshold concentration, whereas MIB only 27% exceeded the other threshold concentration. Come on. So this is what you can see here. I don't want to switch. This line represents the other threshold concentration. So everything beyond is potentially detected by sensitive consumers. And regarding the fractionation of these compounds, what's interesting is that the geosmin is roughly 50% either dissolved or intracellular, whereas MIB, is almost 90% in dissolved fraction. And this is what you see here. So MIB is mostly dissolved. So it's, for me, it's important information to show. This chart is slightly different. So again, top represents geosmin, and the bottom chart, the keyboard is very sensitive. The dark areas represent the raw water samples, so the raw water concentration of either compounds, geosmin or MIB, and the light shaded area represent the concentration in the plant effluent. So as you can see on these charts, so when we have bad taste and odor issues or concentrations, sometimes the compounds go through the water treatment train, and we have some detection in the finished water, and this is the source of complaints from customers. consumers. And these red arrows, I'm not touching the keyboard anymore. Yes, did automatically. These red arrows, just to show, to highlight when the compounds are in the particulate form. So intracellular or particulate bound. The water company there used to call it particulate bound. I like this term. So I'm using it, not very happy with it. It illustrates well sometimes for water treatment perspective. Particulate bound sounds good. So in this case, from 2011 to 2015, the water company experienced a lot of geosmin issues, and when, as a small conclusion of this, so exceedances of other thresholds in finished water occur most of the time when the test and odor compound was in the particulate form, and dissolved form did not generate a lot of complaints from the customers. So I had a look at the removal rates of the plant, because if there's some issues in the finished water, so maybe the water treatment was not so good, but actually it is good. It looks good, actually. And as you can see on this chart, this is the removal rate on the left side is geosmin, on the right side MIB. And we don't have the legends here, but the diameter of each bubbles represent the fraction form of the compound. So large bubbles represent particle bound, and small bubbles or the dots represent the dissolved forms. So independently, the first view of this chart, so independently of the fractionation of the compounds, the removal rate is good, and especially when we have higher concentration in raw water, we have higher removal rates of in the finished water. So the highest concentration you get in the raw water, you have highest removal rates. That is too sensitive. And but as you may, if you carefully look at these charts, sometimes it's not true. It's pain. So it looks like there's a threshold here. I cannot touch. Well, I'm not touching anymore. So imagine there's a vertical line around 10. It's a log scale. So 10 nanograms per liter, 10 ppt. On the left, if you draw a vertical line on the left side of this line, you see that below 10, 15 nanogram per liter, these compounds tend to be dissolved in dissolved form, and higher concentration in the raw water tend to be particulate. So I don't know if it does affect. I didn't play with this. I don't know if it does affect the treatment removal. It may at some point, but here we see that MIB, in the case of MIB, the more particulate it goes, and the most effective is the removal of this compound in the water. So from observations and need to provide solutions to the water producers, and I think it will be interesting for them to develop early warning system, so they can anticipate the appearance, the rise of taste and odor compounds in the water supply, the water entering the plant, before the water enters the plant, so they can adjust the water treatment processes to optimize and in real time, and of course it will be cost effective because they may be able to manage their consumption of organic carbon, for instance. So of course they do routine analysis with the GCMS. So this instrument is very, very sensitive. There are low detection limits, but it's also very expensive and time consuming. There are new technologies, new sensors that are being developed right now. So you may have heard of e-tongs. We had good talks yesterday, a good talk presenting the e-nose. Some manufacturers are developing fluorescent sounds, like the one mentioned on this slide, the BBE fluorescence, but they don't measure the fluorescence directly of taste and odor compounds, the cyanotoxins that they would like to do. They use the free phycocyanin, so they rely on the oxidation processes to disrupt the cell membranes and the release of phycocyanin. Phycocyanin is the photosynthetic pigment of cyanobacteria, which is water soluble. So they use it as a proxy to estimate the release of the cyanotoxin and or taste and odor compounds for intracellular contents of the cells. And there's also other techniques, and I will talk a bit about the fluorescence spectroscopy, the method that I'm trying to use to detect organic matter in the water. So you may have already seen this kind of pictures, or you may not. But basically, we use a fluorescence spectrophotometer. And what the instrument does is it shines some light at a given wavelength. It's switching again, sorry. And depending on the excitation wavelength, we measure the intensity of the fluorescent signal at a higher wavelength, which is the emission wavelength. And because when you shine some light on a molecule, it absorbs some photon energy, and the emitted light in return is also always at a higher wavelength. Not always, but very, very often at a higher wavelength. So it lost some energy, and so the fluorescence signal is measured by the instrument. So some authors identify different regions for the fluorescence matrix. This is a matrix. Now it's a 2D, two-dimensional representation, but you can project the fluorescence intensity on the z-axis, the z-axis in the third dimension. So you get a landscape. Basically, you have bumps and dips and hills and valleys. And so we have some peaks, and the highest intensities area, they are described as peaks, fluorescence peaks. And so one of the author, I'm struggling with the keyboard. One of the author, like Chen in 2003, identified five different regions. So it has a small region 1, for small compounds with one aromatic ring. So it fluoresces like a tyrosine, the small amino acid. The second region is tryptophan, another amino acid, but most of the compounds found in this area, they have a similar fluorescence as these two compounds, 2 rings for tryptophan. And also, if you go to more complex molecules, like region 3, we have more rings and more side chains, aliphatic chains. You go to more fulvic compounds, and also you can increase the complexity of these molecules, and you end up with humic substances. We are very, very complex. We don't know the exact composition of these compounds. But there's also a small area, region 4, that was described as microbial byproducts. And for me, it's not only microbial byproducts, it can be any kind of compounds between humic substances and degraded terrestrial compounds, and also compounds produced by bacteria and or phytoplankton species, algae. So depending on the authors, there are different way to describe those fluorescence peaks. So can we use the fluorescence techniques to detect algal metabolites in the water samples? Some authors try to do that. So back in 2010, Sigman and co-authors they identified a protein-like substance, and they came up with the identification of cyanotoxins. And here I put a picture of a microcystin toxin, which is a polypeptide, heptapeptide, or 7 amino acids. So it could behave like a humic substance. We have many rings, can be complex. Maybe I don't know exactly what was the protein. Maybe it was a cocktail of different compounds, but still, it was the first approach. So they say that maybe cyanotoxin could fluoresce in this area. In 2019, a student, Kan, who worked with Arash Damiadi in his lab in Australia, so she grew algae in batch cultures, and she was able to, she used the fluorescence technique to analyze the culture media of those algal cultures, the medium, sorry. And she ended up with two different response, two different answers, depending the green algae chlorella was found in the region 4, and the cyanobacteria microcystin had a more complex fluorescence. It behaved like humic substances, and maybe it's due to the mucilage surrounding the colonies. Chlorella and microcystis are very small individual cells for chlorella, but microcystis is a colonial cyanobacteria, so you get more cells embedded in the mucilage. So that's why maybe the fluorescence of microcystis species is found in the humic systems, humic area, fluorescence area. And as far as I could go, it's not an exhaustive review of the literature, but in 1992, I found the thesis of PhD thesis of one student, Sharma, at the New Jersey Institute of Technology, and back in 1998, he said, okay, MIB, the 2-methyl isobutyryl fluoresce like tyrosine, so it was found in region 1, and geosmin with these two rings behave like tryptophan. So for me, it's really informative. So because of there's so many compounds in the water samples dissolved in the water matrix, it's highly variable with the season. I joined a program, a research project, which is led by Andrew Tanazap, University of Cambridge in the UK. And he was really interested to track the seasonality of dissolved organic carbon or organic matter in lakes. So I said, oh yeah, that's a good opportunity for me to find out what's in my lake. So I joined this program, and now so the project is running has been running from January this year, and is going to end in December this year. So it's just for 1 year, but he managed to get 45 lakes worldwide. Not all continents are represented, as you can see on the background picture, but we have European, North American, South American lakes, and a few in Oceania. Africa is missing, sadly, because they have a lot of organic matter in their lakes. And so for me, joining this project was a good opportunity to link the dissolved organic carbon is going to analyze using the ultra high resolution mass spec with my algal dynamics in my lake. So I know that I have different algal successions in my study lake. Sometimes we have dominance of diatom, sometimes we have cyanobacteria. It's a classical study case. And but I don't know what's behind my fluorescence peak. So I get fluorescence depending on what algae I get in my lake, but I don't know what's behind. So it will be able to provide hopefully some answers to my questions with his chemical analysis of my water samples. So the goal of today for me is to, in one side, to develop a tool for water managers, water producers to anticipate the appearance of organic matter entering the water treatment plant. And also I will keep analyzing the algal population in my lake and try to figure out what algae produce, what compound they produce. And because, as you can see briefly, it's a small synthetic chart on the right, sorry. When we have a dominance of diatoms, we have a fluorescent signature, fingerprint. I call it fluorescent signature. Can be, it's not specific to, maybe it's specific to diatoms, but when I have a diatom bloom, it's not always 100% of diatoms. It's a mixture of different algal population. So we have maybe 50, 60% of diatoms, but sometimes we have a severe cyanobacterial blooms and we get a different fluorescent signature of cyanobacteria. So when we have a bloom of cyanobacteria, it would be like more easier, but much easier, because we have almost 90% of cyanobacteria, so most of the rest of the algae are outcompeted. So it will be potentially a specific signature of the dominant species. But also, I'm also interested when the bloom crashes, because as you can see on this chart, when we have a depression in the algal dynamics, the fluorescence fingerprint of my water matrix has more peaks, and the fluorescence is more intense. So I'm pretty sure it's linked to higher, I forgot the word, sorry, more intensive release of intracellular contents of the cell. So it can be anything, it will be protein, nucleic acids, smaller compounds, a lot of things. And I don't know what's behind, but if we get more organic matter, subsequently can be an issue for the removal of this organic matter for water producers. That's it. So thank you for your attention. And if you have any questions, I'm pretty sure you do.
Thank you very much, Nico.
Sound.
There's some question on the chat. Sorry, maybe, okay, you can. Yeah.
Thank you for the presentation.
They don't listen.
So we repeat.
Or you can tell them the name. I mean, I know because other people from
Come or you repeat my question.
So now you can talk.
Can talk?
Yes.
You show this tool for a natural water.
Yes.
Can you use it for more engineered systems like water distribution systems? Because microbes are inside, not cyanobacteria, but
yeah, any
microbes
kind
are in the system. They make less amount of organic matter, but can you use it, this tool to,
yeah, I think you can use it anywhere, a different step of the water treatment process, or even after. You can use it at
as a monitoring tool
at the outlet of a wastewater treatment plant, anywhere in your distribution system. I think it's possible. You can do anywhere, basically.
The question is it sensitive or low amount of organic matter?
That's a thing I have to determine, because I don't know yet the exact sensitivity of the instrument. because in natural waters I have a lot of organic matter, so it's not an issue. But for drinking water, or I don't know.
Yeah, but from the other side, it's less noise than the natural water. So maybe there's other trade-offs.
I don't know the sensitivity, but if you have less materials in suspension,
Yeah.
maybe it will behave like pure water.
We'll check it.
Yeah. I may have to at some point if I go to clear water.
Okay, there is some question on the chat, Nico. Three is question. In unfiltered samples, do you assume that you determine total geosmin and MIB?
Do you understand? Because so I'm trying to read the question. In unfiltered samples, do you assume that you determine total geosmin or MIB? Unfiltered, well, that's yeah, I think it's total because we have particulate and dissolved at the same time. Unfiltered, you get everything, and that's why we filtered the samples just to remove the particles and get only the dissolved fraction. That was the point of just to see how if we got a different response. And actually, yeah, unfiltered samples represent total MIB or geosmin, unless there's some evaporation occurring, but I don't think so, because the samples were sealed. Maybe some losses when you open the cap of your bottle, but we try to minimize the losses.
Okay. Second question is Zuhal is asking. Can you read it, Nico?
A question from who?
Zuhal.
Zuhal? Okay.
Zuhal saying, I can read it if not.
So relating next question, do you have any sonication processes before assume the total geosmin? No, there's no sonication prior to the analysis. Basically, the samples were put in a sealed cap vial, and the septum was used directly for GCMS analysis. But no, there's no sonication.
All right. There is Sisi's question. Can you read it? If not,
so is there any comparison studies for fluorescence method for determination of microcystins with other methods of determination of determination of microcystin with other methods of determination LCMS? Not what? No, I'm not aware of the other methods.
I mean, how to compare the results that you have with the fluorescence method with other methods, for example.
Oh, that's one of the future steps, because right now I don't know what's behind any organic compound, not only MIB or geosmin. Maybe it's something we can calibrate and intercalibrate with different instruments, and that would be great if we could do that with GCMS, and maybe we will be able to determine the sensitivity of the instrument as well. Because, so yeah, well, let's do it.
Right.
there is one more question, Nico.
Do you only issue with plant
Asking.
bacteria, or do you also have benthic species? Does it make the detection of TNO of cyanotoxin more difficult? Well, actually, those were pelagic samples. So that's a tricky question, because the dissolved compounds in the pelagic samples may also come from the benthic cyanobacteria. We don't know the individual contribution of the plantic or benthic compartments. No, but assumingly it's total, independently of the source. And so can fluorescence quenching from the matrix cover the presence of specific compound? Yeah, I would say yes. I haven't tested yet. What I showed you was a raw fluorescence signal because it was easier to illustrate for this presentation, but we have to remove, we have to normalize the matrix basically, and we express in Raman units, and you have to get rid, you also have an internal filter. I forgot the term. Internal filter. But to get rid of the internal, I forget the word. Background. I don't know the background light, and there are also, we have to remove a few things like the Raman. I'm losing my. The line, the Raman, and the other. Rayleigh Tyndall, exactly. So this is the first and second order. So we get rid of this. Basically, we don't see it on this last slide. It's removed. It's normalized, but regarding the quenching, it's tricky because I haven't tried it, but most of methods don't deal with the quenching. Sorry, I don't have the answer right now.
I have a question.
Sure.
One of the biggest problems is water level fluctuation because of climate change. It seems to be it will increase in the future. We are getting the highest level of the water level, let's say up to March to April, then decreasing water level. For instance, in Turkish case, people complaining, let's say August, September, mid-November, because the water level really decreasing a lot. That means a lot of benthic cyanobacteria, plenty of cyanobacteria increasing. You have plenty of experience on the lake ecology and reservoir ecology. Do you have any magic solution to stop or dissuade the benthic cyanobacteria increase?
I don't have a solution for that, but I would like to have some. Well, I'm not experienced with Turkish lakes, but because maybe you get your blooms later than we do in France. I don't know. What do you want to know exactly? How can
low level of the water level support cyanobacteria increases, benthic.
Because of drought and water scarcity,
sure,
you have shorter water columns.
Excess of the water level. Sometimes they are decreasing a lot. If you come to the September or November, October, sorry. That means benthic cyanobacteria increasing, give rise to a lot of geosmin ivy. That means what treatment plant seems to be have the solution for the complaining people. Yeah.
Well, it makes sense, but I don't know how to solve your problem because what I'm trying to tell today is to develop a method to detect those compounds in the water.
Yeah.
But the source of the problem, I don't know to solve it. I think it could be a good monitoring tool.
Yeah.
But how to prevent the algae from growing or releasing these compounds? It's another. Problem. Yes.
When you do fluorescence excitation emission spectrum for different samples.
Louder, please.
For the excitation emission spectrum for different samples, do you look at the concentration of, for example, organics? Before you do that, you sort of have a
no, I don't.
Variable?
No. My first objective was to being able to identify algal community changes with the fluorescence instrument, and to know what's behind in terms of organic compounds. That's why I joined Tannenzap's group because it's going to run my samples for characterize the organic matter.
So it's basically it's going to be all those 5 fractions that you have, and you're going to do the concentration, right?
Say again?
So the 5 fractions in the excitation emission matrix spectra that you have, unique acid, fulvic acid, soluble microbial products, etc. So what I'm basically asking is that the concentration of these compounds would also be known. For example, if you have fluorescence regional integration technique, so you can basically know what that fluorescence corresponds to in terms of the concentration of that particular fraction.
Okay, so you have to cross different techniques to make sure, to be confident in what you get at the end.
Yeah, and then you can have the concentration of these different fractions. And if you do, for example, more analysis, I don't know about this SCOCD, liquid chromatography organic carbon detection, and then you can know much more detail about the.
Okay, so okay, yeah.
So I think. So yeah, I'm excited to see these results about fluorescence excitation.
But we have to talk because I
think to sort of do it in a quantitative manner would provide you much more detailed information.
Okay.
Maybe something like
good to know. Well, I'm not a chemist, so I'm not familiar with the different analysis techniques.
Yeah, and again, going back to that crunching, self-crunching, if you have different concentrations of organics in different samples, it will make it very difficult to compare. Just simply looking at the excitation emission exercise.
Yeah.
So if you have a comparable concentration of organics before you do the FEM or these factors.
Okay, good to know. Thank you. Any more questions? All right. Thank you.